So tonight's video is going to cover biotechnology, how we, cut DNA, analyze DNA, make comparisons to other organisms, to other humans, how the actual experiments where bacteria are able to incorporate new DNA into their cells. So we'll look at a wide variety of things in this video. So the first technique we're going to look at is electrophoresis. And electrophoresis, a lot of you are probably familiar with from from TV shows, from either, medical dramas or in particular, like crime dramas. You see the DNA analysis. And most of you have probably seen these gels that look like this, where they have the stripes or the banding from the DNA moving through them. That is all electrophoresis. So electrophoresis is going to separate DNA fragments or DNA molecules according to size and charge. And by charge, we mean actual like positive and negative charges. And so to do this, the first step in doing this is to take the genetic material and to cut it into fragments. And we'll cut it into fragments using these restriction enzymes. So restriction enzymes will cut the DNA at a particular spot. Okay? They'll look for a particular pattern within the DNA, and they will go ahead and they will cut the DNA. And what they will leave behind are what are called sticky ends. So when the restriction enzyme cuts the DNA, it will come through and it will cut between two nucleotides, then it will go between the two strands, and then between two more nucleotides. So you see how it cut here kind of in a zigzag pattern. And so by cutting in this zigzag pattern, it ends up leaving these overhanging pieces of DNA here. So the other one would look like this. So it ends up leaving these overhanging pieces of DNA here. So these are called the sticky ends. And these will become important like a little bit later in certain other things. Okay, but our restriction enzymes are what are going to cut the DNA, and they're going to cut the DNA at a particular location. And we have different restriction enzymes that will cut at different locations. So after the DNA is cut into fragments or cut into pieces by these restriction enzymes, the pieces are then run through a gel using the electric current. So if you look at your picture down here, you'll see you've got this end here, kind of along the top, is where they put the DNA in. And you'll notice that side is negatively charged and the other side is positively charged. Well, DNA itself has an overall negative charge. And if you remember, negative doesn't like negative, but negative and positive like each other. So when we turn the power on in the gel electrophoresis machine, it'll actually encourage the DNA to move towards the positive side and away from the negative side. And that's how we see it start to move through the gel. So we cut it using the restriction enzymes. We run it through the gel using this electrical current, so the DNA gets moved towards the positive end. And the smaller the pieces are, the farther they will travel in the gel. So as we look at this last picture over here on the right, you'll see the shorter molecules are the farthest away from where it was originally loaded into the well, and the longer fragments or pieces are closer. They did not move quite as far. Again, it's moving through this gel electrophoresis. And so because it's moving through this gel, these shorter pieces are going to be able to move through the gel a little bit faster. They move a little bit faster, so they go a little bit farther, where the larger pieces do not. Okay? And then we can use this technique to compare genetic material. Okay? For instance, there are 3 that were loaded into this gel down here on the right, and we can compare and check for similarities and differences between organisms, between various people, okay? And then we can analyze those comparisons. Okay, so another technique that we use in our biotechnology world is this polymerase chain reaction, shortened down to PCR is what it's most commonly called. A PCR will help amplify the DNA. Basically, what happens with what PCR will do is if we have a small amount of DNA, basically maybe not enough to run through the gel electrophoresis, PCR will help multiply it so that we get lots and lots of copies of it. So then we could do analysis on it a little bit easier. Okay, so it's a method of making DNA copies. And to do PCR, you're going to need an initial template strand. You're going to need the DNA polymerase enzyme. Remember, DNA polymerase enzyme lays down more DNA nucleotides. So you're going to need the DNA polymerase enzyme to help do that. You're going to need nucleotides. You're going to need an energy source. And you're going to need primer. Remember, primer actually starts the DNA replication process going. Okay, so it's basically doing lots and lots of DNA replication in a test tube. So in the test tube itself, you're going to need the initial DNA, the DNA polymerase enzyme, the primer, the nucleotides. Okay, and then you're going to need to denature the DNA. So basically, you're going to need to heat the DNA to above 90 degrees Celsius. And what that will do is it will separate the DNA strands. So we denature it, heat it up enough to separate the DNA strands, and then we anneal the DNA, which is going to cool it down. And it's going to cool it down so it will actually build the DNA. Okay, so it will join with the primers and build the DNA. And that's how we'll get our copies. So our next technique involves bacteria. It's called bacterial transformation. So during bacterial transformation, this is when bacteria will introduce DNA into its cells. Basically, it'll pick up foreign DNA. And so it's a way that the bacteria can increase its variety within the DNA, increase its variation within the DNA. And so it will pick this DNA up that's basically hanging out outside around it. So it will pick up the DNA, and the DNA will have, so basically, the bacteria have special surface proteins on them that are specialized to be able to pick the DNA up. You can see the bacteria here. There's the DNA there that bacteria is going to pick up and incorporate into its own cell. And so we'll bring these bits of chromosomes in that are usually from other bacteria. So again, incorporating variation, because remember, bacteria don't sexually reproduce. They reproduce by binary fission. So they should end up being clones of one another. But as we've talked, we know that genetic variation is very important to species survival. So this is one of the ways that bacteria can do that. So they incorporate these DNA bits into their own chromosomes, which can allow them to express new genes that maybe they didn't have access to before. Okay? It can allow for transformation, so altering their DNA strand there. And it is a form of recombination, right? I can form these recombinations here. The DNA can be incorporated directly into the bacterial genome, or it could exist as a plasmid, which are those separate little circular pieces of DNA that bacteria may have within them that they also can express the genes from. Okay? This can be used to produce things for us. For example, we can have bacteria take up the gene to produce insulin and then harvest the insulin from those bacteria. So because of that universal genetic code, the bacteria can pick up this human gene and still be able to do the transcription and translation of it to produce the insulin protein hormone that we would need to harvest. So this can be used for a variety of ways, and we'll talk about this particular experiment and how this works in class. Okay, so another biotechnology kind of method that we can do within the lab is we can actually determine the sequencing of the DNA. A common method for doing that is what we call the Sanger method. And so it's used to determine the base sequence of DNA. So used to determine those nucleotide sequences within the DNA. It's based off of replication. So basically they trace the replication that occurs. It's a 3-step process. Okay, so basically PCR will need to happen. We'll need to amplify that DNA, make sure there's enough of it, separate it by our gel electrophoresis, and then we can analyze the gel and what the gel tells us to determine the sequence. You guys do not need to know details of these kinds of things. Okay? I'm just kind of exposing you to these variety of techniques, and we'll look at some various experiments that incorporate these in. If you follow your diagram here, you can see you've got a single strand DNA here, and they've labeled it with some of these primers, radioactive primers that they can trace. Okay, and then they perform the gel electrophoresis, and then from there, they can analyze the sequence of the DNA. Okay, and so as many of you are aware, DNA can be used for forensic identification. You see it a lot, right, in our crime shows and that kind of thing, where they're comparing DNA samples from the crime scene with the suspects and the victim. They can compare blood samples to determine if it belongs to a victim or to a suspect. We can do what's called DNA fingerprinting. DNA fingerprinting is the actual comparison of the banding patterns. So when we look at a gel, and this is what I think most people think of when they think of the DNA comparison, is, for instance, looking at the gel here and comparing the fragments and seeing if they match, or, where their differences, because everybody has their own unique patterns. So we can determine who the sample came from. We can also use restriction fragment length polymorphism to help with forensic identification. So RFLP is how that's going to be usually abbreviated, right? So our RFLP can account for differences in DNA between individuals. Okay? The junk DNA accumulates in a lot of differences, and so we can analyze those restriction fragment length polymorphisms. We can analyze these chunks of junk DNA looking for our differences. And then our last technique here is what we call our short tandem repeats. Short tandem repeats are just little chunks of DNA that have really short repeating units. So they're repeating over and over and over again. So if you look at your picture over here on the right, you can see that the first gentleman here has the little AGAT sequence repeating 7 times and 6 times, where the female here has it 4 and 19, and then the last gentleman there has it 12 and 14. So we have different varieties of these short tandem repeats. And so those number of repeats can be different from person to person, right? Can be variable among our individuals. And so an offspring can show similar short tandem repeats based on the alleles inherited from the parents. So this female could pass on this 4 repeat and 19 repeat inheritance to her children. The males could pass on their alleles for the amount of repeats they're going to have to their children. So you can make some relationship determinations that way. Okay, so it does make it helpful at determining identification of people. There is a very low mutation rate with these repeats. Okay, so they stay pretty stable and pretty consistent. And so it helps with human identification. And they're easily amplified by PCR. So they respond well to the PCR technique to help gather a large enough sample to do a quality analysis on. So this is just kind of a quick overview of a variety of techniques, and we'll be kind of picking apart experiments that will use these techniques in class.